Hello, everyone. My name is Chris Mathis. I'm the Chief Commercial Officer at AnaBios. It's my pleasure to be the moderator for today's webinar. And this is the third in a series on pioneering pain research. If you missed the first webinar, you missed a good one. Be sure to download this at our website. Dr. Ted Price spoke on human dorsal root ganglion transcriptomics. And then last week, we had an excellent webinar presented by Dr. Cheryl Stuckey, Neuroscience Director at the Medical College of Wisconsin, on sickle cell disease pain mechanisms. And then today, before I introduce our speaker, I just have a few things to say. I'd like to thank AnaBios' Director of Marketing, Gary Watkins, for organizing this series and all of the webinars that we've been putting on since last year. Just a reminder, there will be time for questions at the end. You can enter them over in the right corner on the control panel. Or you can raise your hand if you would like, and if we have time to get to you, we will allow you to ask your question verbally. So AnaBios has put these webinars on in an attempt to bring us all together. During these times of no conferences, several pain conferences have been canceled. We have felt that it's important to continue the dialogue. And so just as there are many different types of pain, we've had different speakers, and we're really excited to hear from our speaker today. Before I do the introduction, I would like to tell you about AnaBios for those of you that are new to us or we're new to you. AnaBios is a unique contract research organization and biotech company. We're located in San Diego. We recover human tissue samples from ethically consented donors and use the tissue or cells to perform physiological assays in which we can test preclinical compounds for drug discovery projects. The fact that we are able to do physiology experiments really attests to the quality of these cells and tissues. So AnaBios is essentially redefined first in human. So if you'd like more information about AnaBios, please send me a message or reach out to us on our website at anabios.com. So again, it's my pleasure to introduce our speaker today. The overall objective of Dr. Stone's research program is to improve diagnosis and treatment of chronic pain by expanding our understanding of its neurochemistry and neuropathology. She received her PhD at the University of Minnesota in 1999. As a postdoctoral trainee at the Oregon Health and Sciences University, she was the first recipient of the John J. Bonica Postdoctoral Training Fellowship from the International Association for the Study of Pain. Following a brief interlude in biotech, she returned to academia in 2002. Since that time, she has received 2 NIH R221 grants and a CIHR operating grant as principal investigator. Dr. Stone is an inventor on 7 patents, has co-authored over 20 manuscripts, and has been recognized for early career scholarship by the American Pain Society. Dr. Stone joined the Faculty of Dentistry at McGill University in 2007, and recently she's come full circle, returning to the University of Minnesota as Professor of Anesthesiology. The title of her presentation today is Chronic Low Back Pain and Epigenetics, Perspectives from Human and Animal Studies. Dr. Stone, take it away.
Hi, good morning. Thank you very much for your invitation to be part of this series. My colleagues, Dr. Price and Stuckey, are 2 of the best researchers and speakers in the game, so it's really an honor to be part of a series with them. I'd also like to thank everyone who's attending this morning. So it's no surprise to this audience that chronic pain has a huge cost to society. One of the best examples of this comes from the Global Burden of Disease studies. These are funded by the Bill and Melinda Gates Foundation, and they track all communicable and non-communicable disorders and diseases in over 300 countries internationally. And in 2013, they completed a study that was published in the Lancet where they evaluated the top causes of years lived with disability. And this is the top 10 globally. So you'll see number 1 is low back pain. Number 4 is neck pain. 6 is migraine. And other musculoskeletal disorders, which is primarily arthritis, rounds out the top 10. So that's 1, 2, 3, 4 of the top 10 causes of disability are pain related. These are not just in the developing world, it's also the case for the developing world, where back pain is still the #1 cause. The only difference between developing and developed worlds in this list is iron deficiency is not in the top 10 in the developed world. So back pain is not just about us sitting at our computers. I like to use this slide to illustrate some of the common causes of low back pain, because the causes are basically everything. So the vast majority of the time, we have really no idea what is causing an individual's pain. This is sometimes referred to as non-specific low back pain. And it's probably about 85% of the time that we're really not sure. In some cases, we do know that there are links to spinal structures. So I'll get into that in a moment. But there's also the sensory nervous system and the spinal cord and how that input gets processed, how the spinal cord sends information to the brain, how the brain adapts to chronic painful input, as well as the rest of the body, this other, the immune system, the GI tract, cardiovascular system, systemic inflammation, and so on. So I'm going to touch a little bit on all 4 of these areas in the presentation today, starting with the spinal structures. So we know, depending on who you talk to, maybe 10 to 20% of the time, that there is a link between low back pain and intervertebral disc degeneration. However, this link is kind of tenuous, because if you take populations, there's a very poor correlation between disc degeneration and pain. And there's a couple of reasons for this. The first reason is that the percentage of adults in the general population that have disc degeneration is approximately equivalent to the age. So if you have a room full of individuals that are 50, probably about half will have at least some moderate disc degeneration. But the vast majority of those individuals are not experiencing back pain. The other reason for the low correlation is because of the many other causes of back pain besides disc degeneration. There's other structures in the spine, there's joints, ligaments, tendons, spinal muscles, and all of those things feed into the state of the individual, their own individual genetic and environmental risk factors, and so on. So the first question that we're going to ask today is why the slide, there we go. Okay, so why is disc degeneration painful in some individuals but not others? So this is a huge question in the field, and it's hoped that if we can answer this question, provide insight into this, maybe we can use that information to help those who are not so lucky. So in the first study I'm going to describe, we took an approach where we used human spinal fluid to do discovery-driven screens to try to understand what might be different between these 2 populations. So we recruited individuals that either had low back pain, chronic low back pain, or controls. And the individuals with chronic low back pain were scheduled for spinal surgery, and they had severe disc degeneration of at least 1 disc. We then collected spinal fluid from all of the participants, and we sent everybody for a lumbar MRI, which allowed us, including the control asymptomatic participants, because that allowed us to separate the samples into 3 distinct groups. So we had a group with back pain and confirmed disc degeneration. We had a group with no pain and no moderate or severe disc degeneration. But then there was a group that did not have pain, but did have at least moderate disc degeneration. And by comparing specifically the pain and the no pain groups, we can ask this question: what's different between those that have pain and those that do not? Just to reiterate the mismatch here between degeneration and pain, the code here is low back pain and DD is disc degeneration. So these are the MRI scores where the controls who have minus low back pain, so no pain, were split into individuals that had no discs that were graded at least 3 or more on this 5-point scale based on MRI. And then we had the asymptomatic participants with disc degeneration and no pain. And you can clearly see the mismatch between the pain patients who report high pain intensity and the control subjects who do not, regardless of the degree of degeneration. We then conducted some discovery-driven screens in the CSF, both using proteomics approaches and some hypothesis-driven cytokine screens. And one of the most interesting things that we found in these screens was interleukin-8 as being uniquely upregulated in low back pain, but not in the asymptomatic controls. So we confirmed that in the human CSF with ELISA assays. So this is the concentration on the y-axis and the 3 groups, and you can see the increase in the group with pain. And we also looked at the intervertebral discs, because for many of these participants who had pain, we were able to obtain disc samples during their surgery if they had back surgery. So we actually have also shown that some of this IL-8 may be coming from the discs. So the question here, of course, is this a potential therapeutic target, and how do we address that question as a proof of concept? So this brings us to the role of preclinical models and the power of integrating human tissue studies with preclinical models. So we've been working for a while with a preclinical model of back pain associated with disc degeneration. In these mice, they're missing a gene called SPARC, which has a horrible name. It's called secreted protein acidic rich in cysteine. SPARC is expressed in intervertebral discs. It is decreased in discs that are degenerating and painful. And in the mice, the deletion of SPARC results in a progressive age-dependent disc degeneration that is associated with behavioral signs of axial discomfort and radiating leg pain. So it is, one thing that is nice about this model is that the progression of disc degeneration happens over time. So at 6 months of age, the SPARC null mice might have a few discs that are moderately degenerated, and another mouse might have one severely degenerated disc. And as they age, it gets progressively more likely that a given disc will be degenerated and the severity increases. So it's very much a naturalistic model of the evolution of disc degeneration that happens with normal aging in humans. So we wanted to ask the question, does blocking IL-8 in SPARC null mice attenuate signs of pain? Before we get to that, I just want to show you in this model that we also are able to demonstrate increase in IL-8 just in the SPARC mice compared to wild type. So what you're looking at here is the full difference in expression of proteins that are released by the discs ex vivo in the culture system. So we excise the disc, culture it for 48 hours, and then take the supernatant. And this gives us some insight into what those discs are releasing. This approach was pioneered by our collaborator, Dr. Elizabeth Haglund, and she has done a lot of fascinating work using this approach with excised human discs from tissue donors. So what I'd like to point out here is simply that the mouse IL-8 homologs, particularly this one, LIX, is in fact also upregulated in degenerating spark discs. So the behavioral model that we're using, in this case, I'm going to show you a sign of radiating pain. In order to measure radiating pain, we test the hindpaws of the mice. And what we found in our experience with these animals is that they develop a very reliable hypersensitivity to cold in their hindpaws. So what you see here is how much behavior they produce when we put a drop of acetone on the hind paw. And animals that are sensitive to pain will shake their paw or lick their paw when the acetone evaporates because it creates a cooling sensation. So here in our wild type animals, you see they're less responsive. Acetone evokes fewer behaviors than the spark mice. One thing that's really important for translation of preclinical studies to the clinic is that we use routes of administration and time courses that are in line with the clinical situation. So in this study, we gave the treatment, which is a drug called Reparexin, 3 times a week, systemically for a total of 8 weeks. And what happened was, if you follow the blue line here, it took more than 4 weeks, starting about 6 weeks, and then really coming to a complete attenuation at 8 weeks for this drug to have its effect. So I want to make a small plug here for doing long-term studies with drugs, because we may have missed a lot of really powerful pain medications preclinically by only testing an hour later or 24 hours later. In addition to having efficacy in the mouse model, which provides proof of concept, we also looked at the discs from the mice that were treated. And this is the slide you saw before comparing wild type versus spark degenerated discs. When we took the discs after the treatment period and did the same experiment, comparing spark null vehicle treated to drug treated, we actually see a really nice decrease in many of these pro-inflammatory cytokines, including the IL-8 homologs and interleukin 1 alpha, beta, and 2. For this work, the future directions, one of the big ones is to determine which of these mediators, if it's IL-8 or another cytokine or maybe other pro-inflammatory or pro-nociceptive mediators that are expressed in degenerating discs, what is their direct effect on sensory neurons? And this is a really important direction that we want to go in, because the discs are going to become, as they degenerate, increasingly toxic. And if we can kind of slow that down or block the effects of the inflammation, the discs on the sensory neurons, we might be able to intervene. Okay, so the next example is coming from sensory neurons. And this is a picture, schematic of a nice healthy disc. The inner part has this jelly-like substance, and then it has cartilaginous layers around it. And in a healthy disc, there's very, very little nerve and vascular ingrowth, and it only goes into the outer parts of the disc. So normally, chemicals in the disc pH in the disc, mechanical forces in the disc are not normally sensed by the nervous system. In contrast, as discs degenerate, they become increasingly innervated and increasingly vascularized. So the question is this potentially driving pain in some individuals? We have been looking at the innervation of painful human discs, trying to understand what kind of fibers are growing in and if they might be the source of pain from those discs. So this is an example of innervation surrounding this is a blood vessel, and PGP is a general marker for nerve fibers. And this was taken from fairly deep into a human disc that was removed surgically. So these fibers should not be there, but they are. And immediately it creates a mechanism by which the contents of the disc could activate these fibers. To determine if they may in fact be nociceptors, in this example, we co-stained with substance P, which is a marker for one class of nociceptors. And in fact, there are some substance P fibers in there. So we wanted to know what might be driving this nerve ingrowth. And is it generalizable across all degrees of disc degeneration? So when surgeons are considering whether to perform surgery in a person with back pain, they're going to do an MRI and they're going to look at the discs and grade them according to this 5-point scale usually. And the discs that are moderately to severely degenerated are thought to be the ones that are most painful and the most likely candidates for surgical removal. So what we did here was we asked the question, is nerve growth factor, which would cause this ingrowth, upregulated in discs? At what degree of degeneration is it upregulated? And it was very interesting to find that the highest peak is in the moderate to severely degenerated and not in the most severe. Thinking about it more, that's making sense because very severely degenerated discs are often acellular because the cells have just died. So it's possible that these are the discs that are really the most painful and have the most nerve ingrowth. So is this a potential therapeutic target? Not just nerve growth factor, but the whole pathological innervation as well. So the first experiment that we did was testing anti-nerve growth factor in the SPARC model. So in this case, we're still measuring cold sensitivity, but it's on a cold plate. So we see that wild type mice take longer to withdraw their paws from the cold plate than the SPARC mice. So that confirms the presence of cold sensitivity. And we followed a protocol already in the literature and gave anti-NGF antibodies, IP, 2 weeks apart, twice 2 weeks apart, and saw basically a complete reversal of cold sensitivity in the NGF treated animals. So this is very exciting because, as I'm sure most people in the audience know, there's anti-NGF therapies are in clinical development and have been for a long time, and may be one of the most exciting things that's happened in pain management in a long time if they're approved. But it seems unlikely that this is going to be the first line target for someone who's developing back pain. What is the first line target is almost always not pharmacological. So these are treatment guidelines put out in 2007, and all of the interventions that are in the red boxes are not pharmacological. So we have acupuncture, cognitive behavioral therapy, exercise, spinal manipulation, massage, yoga, all of which have some evidence for efficacy. And the very first thing that is recommended for someone with a new case of back pain is to keep moving. So we were really curious what would happen if we gave the Spinal mice voluntary home cage running wheels. So this is a really low-tech intervention. They have these little igloos that they build their nests in, and the wheel spins and the mice love it. And when you go into the animal room, you just hear vroom, 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 because they're just all running on their wheels. The control group is the same thing, the hut and the wheel, but we put a little screw in it so that the wheel doesn't spin. So they can still nest, they can still play on the wheel, but they don't get the voluntary running. And what we found when we just put these little plastic things in their cages is that over time, it reverses cold sensitivity. So here's the wild type animals being less sensitive to acetone than the spark mice. And 2 weeks of wheels, not much, but by a month, 2 months, we start to see a real effect. Interestingly, the older the animals, the more severe the degeneration and the longer this running intervention takes to have effects. So in this particular experiment was done in mice that were, I think maybe 3 to 4 months old at the beginning. If we started in mice that are closer to 10 months old, it takes 2 to 4 months to see efficacy. And that's consistent with the degree of pathology and what's needed to reproduce or to reverse it. So this was really exciting. And we also have done experiments that suggest that this is not a runner's high acute opioid release effect. Because if we take the wheels away, it takes 1 to 2 months for the pain to return. So this is a disease-modifying intervention. When we looked at the effect of running on innervation in the discs, you can see here that comparing the wild types to the Spinal controls, Spinal mice have a big increase in innervation in their discs. So that would have been predicted from the human stuff also. But look what happens in the animals that had home cage running wheels. This is just the effect of increased physical activity, and we almost completely abolish the pathological innervation in these discs. So some of our future directions in this area are to really dig into the molecular mechanisms that underlie these beneficial effects of exercise in discs, but also in other tissues as well. We're also really curious from a pharmacological perspective if increased physical activity or diet or other lifestyle changes can have beneficial effects on the efficacy of drugs that we use for pain. So for example, if somebody is on opioids, would they be able to reduce their doses if they were also more active? Perhaps even there's a synergistic interaction between these lifestyle interventions and pharmacological treatments. Okay. So we're going to talk about the brain now, but before that, we have to take a detour. And the detour is called, what is epigenetics and why should I care? And I hope that by the end of this presentation that you have a better sense for both of these for answers to both of these questions. So epigenetics literally means on or around genetics or genomics. And it is a term that is used to describe all sorts of modifications that change gene expression without changing the genetic code. So in this schematic, we have two identical cells. And in response to the experience of these cells, their environment, what chemicals they're exposed to, what growth factors they're exposed to, they may differentiate into having two distinct phenotypes. Just like in us, we have the same DNA in all our cells, but neurons are quite different from immune cells, which are quite different from keratinocytes and so on. It's all about with the pattern of genes that gets turned on and off based on the environment of that organism or that cell. Some of the common, the fundamental molecular mechanisms include DNA methylation, histone modifications. So in this schematic, we have chromosomes, which are the DNAs tightly packed around these things called histones. It wraps around them. And in order for a gene to be transcribed, the histones need to allow the region of DNA that needs to be transcribed to become available. So these histone modifications, acetylation, methylation, they control the opening and closing of parts of the DNA that's needed, and in that sense, they can control the degree of expression. If a gene is all wrapped up tight and there's no access to it, it won't be expressed. If that is loosened, it's more likely to be expressed. In addition, there's chemical modifications that are made directly to the DNA, specifically the addition of methyl groups to CG sites. And this has several, this impacts the expression in several ways. Usually, although not always, but usually it causes a decrease in expression because the methyl group will block the binding of transcription factors. But it's not always that simple, of course. And to give a sense for the power of these epigenetic processes and their ability to really shape the organism, I'm going to give you an example from the work of Sif and Nini in Montreal. So what they've been studying is the impact of early life experience. And in this example, they have rat mothers that either provide a lot of maternal care for their pups, which includes quite a bit of licking and grooming for the pups, or mothers that tend to give less maternal care. And what they found is that animals that had a really rich early life environment have a decreased stress response when they're adults, and they have increased resilience in the face of novel situations. In contrast, early life deficits will lead to increased stress responses and decreased resilience in the adults. And sparing you many, many molecular studies that demonstrate that, in fact, this experience or environment early in life imprints a behavioral impact in adulthood. And this team has been able to demonstrate in the that the HPA axis is actually dysregulated by the impact of methylation because of your early childhood experience. So something that happened to you a long, long time ago may still be embedded in your genes and controlling expression in systems that then can set how you respond to a certain situation that you're challenged with later. So now we will go back to the brain. It had been shown a while ago now that participants with chronic pain have altered brain anatomy and function in some cases. And there's been several studies now that have shown that interventions can help attenuate those changes. So we hypothesized if the brain is plastic, can be influenced by the chronic painful input, and if it can also be influenced by the removal of that input, could that possibly be mediated at the epigenetic level? So in order to address this, we first went back to the animal models and we performed a nerve injury, the spared nerve injury, for those who are curious, and measured in the frontal cortex just the total amount of methylation. And we found a difference between total methylation in the cortex of injured animals compared to controls. So that inspired us to do a much deeper experiment in rat frontal cortex, very late after nerve injury. So this is 9 months after nerve injury, where the pain condition is deeply embedded. And what you're looking at here is a heat map. Each row is an individual gene, and each column is an individual rat. And what we did was we asked the computer to separate, based on the pattern of methylation in each animal, could they separate the patterns into distinct groups? And the separation is nearly perfect. The unbiased clustering algorithm was able to identify and separate the sham animals from the nerve injured animals, which is pretty amazing because the only difference in the lives of these animals, they're genetically identical, their food, their environment, everything is the same. It's just whether or not they had a nerve injury 9 months ago. And this degree of clustering is so profound that it led to the following quote from Renaud Massart, who is the first author on this paper. He said, "This kind of clustering is relatively rare. They usually obtain it, this kind of clustering, by comparing different tissues or species. So the brain of these rats has become so different as to be more reminiscent of a completely different tissue or tissues from completely different species," which is a really powerful illustration of the potential impact that chronic pain can have on an individual. We were curious how long these changes take to develop. So this is new work that's just in press currently. And Lucas Topham, in collaboration with many others in the lab, conducted an experiment where we collected the frontal cortex from mice 1 day, 2 weeks, 6 months, and 1 year after nerve injury. And asked the question, so are the functional differences the same across time? Do they occur quickly at 1 day, or do they take months to develop? And what we found was that even 1 day post-injury, there was huge changes in the frontal cortex epigenetically. However, the pattern of these changes and what gene ontologies, gene functional categories they relate to is completely different as the pain evolves. So these are a few examples of some gene ontologies that were common throughout at least 3 of the 4 time points. There was a lot of cytoskeleton coming up in these screens, quite a bit of T cell work. So it's really important to think about the timing of the intervention. So if we're intervening 2 weeks after nerve injury, the molecular pathways that would best be targeted may be very different than if we're intervening a year later. So the next question, now that we've shown that there are massive and persistent epigenetic changes in the brain in this model of nerve injury, is it possible theoretically to target this? And the approach that we took was to do an experiment with a over-the-counter nutritional supplement called SAM-E. So this picture I took off of the Costco website. So you can buy this at Costco, and it is marketed for mood balance and osteoarthritis pain. SAM-E is a methyl donor that endogenously provides the methyl groups for the methyl transferase that then puts the methyl group on the DNA. So theoretically, by enhancing the amount of SAM in the system, we might be able to encourage more methylation or allow our biology to put the right methyl groups in the right places. So it's a very naturalistic experiment. And we really wanted to model it in a way that people would use the supplement. So we gave the supplement orally 3 times a week for 8 weeks. At the end of that treatment period, you can see here, this is nerve injury. So now we're looking at mechanical threshold. And sham animals that received saline are less sensitive to mechanical stimuli than nerve injured. And chronic treatment with SAM partially reverses that. So it may be possible to target therapeutically these endogenous epigenetic regulatory systems. An important side point to make is in studies like this, I want to encourage everybody to also think about the time after injury before an intervention begins. And in this case, we wait at least 3 months for most of our studies to make sure that the chronic pain has really developed and is embedded. Because we might have very different situation as supported by Lucas's work a few slides ago, depending on when the intervention is done. After this testing was done and we saw that SAM did have some effects, we collected the frontal cortex and asked the question, does chronic SAM treatment have an effect on cortical plasticity in the epigenetic signature in the mice? So it did. So let me walk you through this figure. Each row here is an average of the amount of methylation for a specific site on DNA that may or may not be methylated. And these groups are averages. So this is the average of, I think we had 4 or 5 animals that we sent for genome-wide sequencing in this experiment per group. So Lucas here has arranged them by those that are most differentially methylated, decreased or increased. And if he takes the same genes in the injured vehicle animals, so this is the effect, comparing here to here, this is the effect of nerve injury. You see a very different pattern, and just visually, not even thinking about what the individual genes and pathways are, just visually you can see that the animals that had the treatment reverted to a signature that is closer to their uninjured counterparts. So we're currently looking into what these genes and pathways are. We're also very interested in looking at the effects of other environmental changes, diet, chronic exercise might have on these cortical pathways, and hoping that this provides more insight into potential molecular targets. Okay, finally, in the last couple minutes, I'm going to show you some newer work that is other. So in this case, other is the immune system and blood. So I showed you earlier the work by Renaud Massart in the rat cortex, where he showed really massive epigenetic dysregulation in nerve injury. In the same study, he also looked at T cells from those animals. And this is just showing the false discovery rate and the number of genes that are differentially methylated depending on where you put your statistics. So there were quite a number of genes that were differentially methylated in T cells in animals that had exact lives except the nerve injury. And further, he was able to show that he could identify 11 individual sites that are methylated in the entire genome. Just 11 sites could predict with some degree of accuracy whether or not an animal was nerve injured or control. So this earlier experiment got us really excited about the idea of epigenetic biomarkers for pain. Because if we could identify in T cells from humans epigenetic signature or a small number of individual sites that are or not methylated to some degree that separates pain and no pain, that might be a really useful biomarker, as well as provide clues into potential interventions. So we asked this question in collaboration with the Quebec Back Pain Consortium, and the preliminary proof of concept data that came from a genome-wide, methylome-wide mapping study of T cells collected from women and men with and without back pain. So we administered some questionnaires, we collected blood and saliva, and isolated the T cells, and we sent the discovery cohort of 32 individuals for sequencing. And these are the heat maps that came out of that. So let me just walk you through what we have here are the women. These are low back pain cluster. And then there's a control cluster, a low back pain cluster, and a control cluster. And particularly, these low back pain are very different than these controls. In the men, it was a little bit more clear, and they actually clustered fairly cleanly between back pain, individuals with back pain, and individuals that did not have back pain. One of the most interesting things from this data so far is the realization that men and women are completely epigenetically distinct. There's virtually no overlap in the over 2,000 individual methylation sites in females that responded to pain compared to the 400 or so males, virtually no overlap. So my collaborator, Dr. Mauricia Sif, who has had a huge role in this epigenetic work, he says that males and females are epigenetically different species. And this kind of data really suggests that that's true and further reinforces the trends towards including both sexes in our studies. In order to try to identify a biomarker, where it's not practical to take the entire genome and do a genome-wide study for every individual. So what we tried to do in this discovery cohort is to identify a small number of individual sites that that were differentially methylated that together could create a polygenic, a multiple component score that could predict whether an individual is has pain or not. So this is the male data, and the colors indicate whether the extent of methylation at each particular site, and each site is a row. So this row, this row, this row. And we can create a polygenic score. And what we basically found when we created the score based on just 3 individual methylation sites from the entire human genome was the ability to fairly accurately predict whether a participant was a control or had low back pain. But this prediction does not extend to females at all. It's completely random in females. And the converse was true. When we analyzed them separately, we created a female polygenic methylation score. It was very effective in predicting female pain, but not at all useful in males. And a very preliminary, a very exciting thing that we've been doing lately is trying to use machine learning techniques to train algorithms, which then might be able to predict whether or not an individual is in the pain or not. So this is very preliminary. I want to reinforce that, and we do not have the sufficient subject size at this point to do this with adequate power. But it is really encouraging that after training an algorithm on a small number of individuals and then giving them blind numbers, we were able to get some separation, a statistically significant amount of separation in the prediction as to whether or not an individual has back pain or not. Very preliminary. So there's a lot of work to do on this potential biomarker front. We need to validate and possibly improve the polygenic methylation scores, looking at the effect of interventions on these patterns, looking at potential epigenetic biomarkers that can predict whether someone with acute pain may become chronic or can help stratify patients into what treatments might be best for them. These are all really exciting directions that we're going in now. And finally, just to kind of wrap everything up, what's really exciting about epigenetics is that it's plastic and can be modified. So there may be factors early in life that preset an individual to be more risk for developing chronic pain after an acute injury or a pathology, or early life events might be a risk factor for the pathology itself. The transition from acute to chronic pain itself changes the epigenetic profiles, both supraspinally and in the blood, and many others have also shown in sensory neurons and in spinal cord epigenetic changes in pain. And then treatments, whether it's pharmaceutical or it's injections or it's physical activity, they will change the epigenetic landscape. And all of that will help hopefully predict and improve outcomes. So I would propose that it may be possible to develop epigenetic biomarkers at many of these points that can then be used to help guide treatment opportunities in the future. And finally, there's many people who have contributed to this work over the years. I'd like to point out Dr. Magdalene Milkamp, who's been the main driver on the preclinical work, and Lisbeth and Emerson, who have contributed extensively to the IL-8 project and the human discs. Dr. Marcia Sif, who is our epigenetics collaborator, and the team at the University of Minnesota that contributed to the collection of the CSF and the surgical discs. So with that, I will be happy to take any questions.
Thank you very much, Laura. Fascinating presentation. Really emphasizes the importance of looking at the differences between males and females in pain research. And I must say also, it seems like really good news for runners, those of us with nurturing mothers, and Costco members. So very, very interesting. So we have time for questions. They are starting to come in. I'll get things started as people write them in. Again, if you have a question, you can write it over in the right side. You click on the questions tab and write it in there. So in the IL-8 studies, you mentioned future direction is to look at direct effects on human DRGs. How about human spinal cord? Is that another area of interest? Seems to me that might be affected.
Yes, absolutely. We know very little about changes in the spinal cord in back pain. There's been a lot of work in nerve injury, but very little in back pain. We do know that there is some neuroinflammation in the spine in the Spark mice. They have increased astrocyte activation, and they have an increase in microglia, I believe. This is just by immunohistochemistry. So it would be really fascinating to start to understand the changes in the spinal cord that are occurring in individuals with back pain and what we can do to potentially reverse them.
Interesting. Yeah, we'll have to stay in touch because AnaBios is developing human spinal cord assays, and so that might be useful for that kind of research. So here's a question from Cheryl Stuckey, our speaker from last week. Is IL-8 increased in the CSF of human patients with chronic low back pain? Do you know that yet?
Yes. So the IL-8, the original, the reason why we focused on IL-8 was because in our discovery-driven screens, it was increased in the CSF in individuals with back pain.
Okay. Here's another comment and questions. Great talk. Have you looked at the cortex epigenetic signature and its reversal by SAM and other mouse models of pain?
So far, we've only used the nerve injury model. So that's a great point. We really need to understand the cortical impact of other models as well. And in particular, to make any of this useful clinically, back pain is a really mixed bag. So there's neuropathic components, there's inflammatory components, nociceptive components. So it'll be really important to extend this work to other models and to understand the commonalities across different pain conditions. The other half of the question was about SAM. So we have found cortical changes in SNI in nerve injured animals that are reversed with SAM. We're currently digging into that, what genes they are, what functional pathways they belong to. We're also working with a data set of SNI from SNI nerve injured animals that had chronic exercise. So these animals they had pain for at least 6 months, and then they had access to running wheels. Their mechanical hypersensitivity was attenuated by the running. And in our preliminary analysis, it does look like that exercise program is also going to help reset the cortical epigenetic profile.
Okay. The same person asked, have you looked at the T-cell epigenetic signature in other human pain disorders?
No, we have not, and we really want to do that work. And again, for the same reason, what's common across pain conditions and which might make the best targets, but also what's different, because then maybe we can use these as biomarkers to help match individuals to the best treatment. We have not done that yet.
One of the downsides of a virtual presentation is you can't hear the applause at the end. Well, there's a comment from one of the attendees. Very informative. You presented the subject as art of science. Thank you.
Thank you.
Nice. Here's a question. What gender did you use in your rodent studies?
Yes, excellent question.
Yeah.
So almost all of the rodent work that I showed was in males. particularly epigenetic work, we've only done in male rodents so far. So that's another really important area for future direction, is to repeat and extend all of those in both sexes.
Okay. Here's a question about the IL-8 studies. Why did you focus just on IL-8? There seems to be a lot of cytokines differentially regulated between disc degeneration and pain without disc degeneration.
Yeah, that's also a great question. And IL-8 was one of the few things that we found that was significantly higher in individuals with pain and disc degeneration compared to individuals with asymptomatic disc degeneration, so those that had degeneration without pain. So we chose to focus on it for that reason. But what we've been learning in the course of doing these experiments is that there may be many different ways that we can target these inflammatory cytokines. And perhaps you'll recall the study where we looked at changes after the chronic blockade of the IL-8 signaling. And it wasn't just the IL-8 expression of IL-8 homologs that was changed, but many other pro-inflammatory cytokines were also decreased. So any number of those, or things that we haven't even identified yet, might be really excellent targets.
Right, absolutely. Here's another question. Are all the 11 CPG sites uniformly methylated, or are some methylated and some demethylated that contributes or causes or can predict the pain phenotype?
So I apologize that I do not remember.
Okay.
But the important thing to keep in mind is that it's the difference in methylation status between the two groups that matters.
Right.
Yeah, that should be in the paper if the if anyone wants to look.
Okay. Another question, will you stratify the epigenetic study separating patients with disc degeneration but no pain versus patients with disc degeneration and pain, similar to your first study?
That is a great idea. Unfortunately, from that particular cohort, we did not have the foresight to collect blood. But that is definitely a really important future direction.
Okay, and the same person asked, have you looked at patients treated with opioids versus opioid naive patients?
No, that is also an excellent study. I would be willing to bet that there is going to be an epigenetic signature in the blood in opioid users. And it will be very interesting to also see how much that overlaps with the pain signatures.
Yep. Here's a question about translation. Are the epigenetic signatures seen in humans mirrored in rodents? And does this further reduce the translatability of animal studies?
Ooh. So I can tell you what we do know, which is that when we compared in the studies in the mouse brain, the epigenetic patterns, the individual genes, and we looked for overlap with individual genes that are known pain mediators. So we used Jeff Mogel's pain genes database as well as Luda Dyachenko's human pain genes database. And there was a disappointing amount of overlap. So it does raise questions as to whether looking at individual specific genes and gene subtypes will be highly translatable, but I do believe that there are organizing principles in biology and that there'll be much better overlap between the pathways and the general responses in animals and humans.
Okay. The questions keep coming in. Are you able to stay on for time?
Yes.
Go through some more questions. And those of you that have to leave, you can always come back and listen to the recording. So this is a comment and a question. Great talk. Interesting you saw male-female pain signatures that were so different. Are you aware of any work on dogs with intervertebral disc disease? Would this be a useful model for your studies?
It would be a very useful model. Dogs are commonly used in orthopedic studies. companion animal studies as well as some experimental models. And it would be a great intermediate between the rodent work and the human work.
Yeah.
There is there is some movement towards that in the field. Yeah.
Okay. Another comment and question. Great talk. Do you have evidence that the mode of exercise is important for pain reduction? And secondly, do you expect that there will be a sex-related difference?
Oh,
mode
great.
of exercise.
Great questions.
Yeah.
So the first question about the mode of exercise, we have no idea from the rodent studies because you're kind of limited on what you can do there. They can swim, which stresses them out, so we've avoided.
Right.
There's forced treadmill exercise, which stresses, so we avoided it. So we don't know if this is specific to aerobic exercise or if it's just any kind of increased physical activity. There is a big literature in the physical therapy world about aerobic exercise being beneficial as well as non-aerobic physical activity like yoga. So it might be the joke is that the most effective form of physical activity is the one that you will do. Right,
exactly. Well, it seems to me that swimming would be good. It's not, there's no impact, and I understand the limitation with rodents, but that might be interesting to study with humans.
Yeah, very much. And yes, I'll let it.
So here's a question. Do you think that male-female epigenetic differences are primarily driven by sex hormones per se or other environmental factors? Any idea there?
I have no idea. Great question. But I have no idea. Yeah. If I could guess that, like most things in biology, when we ask either or questions, the answer is normally both.
Right. Okay, we'll take a couple more questions. So here's a comment or question. Thank you for a great talk. Have you sorted epigenetic changes to T-cell subtypes?
We have not dug that deep. That's a great question. Well, one of the directions we're considering going in is kind of the opposite of that, which is to start exploring saliva as a tool for epigenetic biomarkers because of the utility of it. So you can imagine a saliva swab and a small kit that can be done on site or in any lab anywhere that could, looking at a bunch of epigenetic markers could help predict or diagnose, predict outcomes or diagnose. So the trick is how deep do we go into something like the different kinds of cells versus a much more clinical approach where it's really about practicality. Both are so interesting.
Okay, one more. Do you believe that back pain progression may have a clinical marker one day that may replace subjective 1 to 10 scales and may give us a more individualized model of prescribing therapeutics as such?
I don't think we're ever going to replace 1 to 10 because in the end, what we care about is how that person feels.
Right.
And whether they have pain or not. But I do believe that epigenetic markers could help predict risk for developing pain and help identify and target better treatments to the right people.
All right. Well, on that, Laura, thank you so much for this great presentation. For those of you on the line that are wondering about the recording, we will post that and send an email with the download. You can watch on demand. And if you didn't get to see the recent webinars on pain that we provided by Dr. Price and Dr. Stuckey, you can also view those. So thanks to all of you who attended. Thanks again to Gary Watkins for putting this on, and hope you all have a great day. Thanks again, Laura. Take care.
Thank you.